and that term is hematocrit. Hematocrit is the examination by percent of the blood being formed by the formed elements. And so what we do is we determine hematocrit based off of what's referred to as a hematocrit tube. We take a small little tube of blood, we put it into the centrifuge, and we spin it. And what we end up getting something that looks like what we see here, where we're able to measure the amount of RBC and buffy coat. So within the buffy coat, we'll see the platelets and the leukocytes. The amount of formed elements relative to the amount of plasma that happens to be there. And we're going to represent it based off of a percent within the total amount of blood that's there. So the formed elements are going to be the cells that we see there, and we're going to compare it to the total blood volume. And we have a typical range of somewhere between 35 and 50% of total blood being formed elements. That's going to vary based off of a number of demographic and physiological issues. Females will be lower in this range than males. Early adaptation to high levels of activity, early adaptations to moving to higher levels of elevation will increase hematocrit. The greater the percentage of cells that we see is an indication of two things. The two things are a sign of dehydration or what's referred to as a polycytotic disease. That is, we have too many erythrocytes in circulation or too many leukocytes in circulation. If we happen to have a lessened percent, it's an indication of anemia or a leukopenic disease. Now, what we have to remember here, regardless of how the books present this, the greatest change that we see in hematocrit is due to shifts of plasma volume more than changes of cell amounts. Because we're doing this as a relationship of cells to total blood volume, if we change the volume, we automatically increase the hematocrit because we're dividing by a smaller number. One of the things that we have to worry about when we're measuring anything within the blood relative to the blood chemistry or the actual cell composition is the hydration status of the individual. And that's because any change in the 90 to 95% that happens to be fluid, water, any change of that amount will automatically change all other measures. And so if we look at this breakdown, this is what we're looking at in terms of the hematocrit and the compositions of the plasma, where the majority of the formed elements will be erythrocytes, followed by platelets, and then finally the leukocytes. And within the leukocytes, the principal leukocyte that we're going to see are the neutrophils, followed by the lymphocytes, followed by the monocytes, followed by the eosinophils, and then finally the basophils. Now we have to remember here is that the change in leukocytes and the change in erythrocytes is going to be based off of immune activation and growth signals based off of physiological stresses. If we were to go about and look at the difference that we see between genders, now remember I asked the question, why would we see the difference between males and females? And this is where we start seeing the differences. So we want to keep hematocrit relatively stable. If I happen to have more erythrocytes and I want to have a relatively stable hematocrit, that means I have to have more total blood volume. So if I'm a male and I have more erythrocytes, that means that I'm automatically going to have more blood volume. Now, I can't get so much blood volume that I dilute the blood, which is why for males, we have a higher hematocrit percentage than we do for females. So notice that we have higher erythrocytes, higher hematocrit, but we also have higher hemoglobin amounts. The higher hemoglobin amounts is directly related to the higher erythrocyte amounts. Now notice with the leukocytes and with the platelets, there's no difference. We don't see a difference in the counts for leukocytes or platelets in between the genders. So let's go ahead and let's move forward here and look at the erythrocyte and what the erythrocyte is going to be doing. So the erythrocyte is going to be carrying the oxyhemoglobin molecule within the central third of the cell. The cell itself is a biconcave cell. This biconcavity of the cell increases the total surface area on the erythrocyte. This increased total surface area on the erythrocyte increases the diffusion principles based off of Fick's law. That increases the rate of exchange of gases in between the erythrocyte and the plasma, so as to allow for transportation of oxygen, which is not very soluble in the plasma, but it also will be involved heavily within the regulation of plasmal pH through the diffusion of carbon dioxide and bicarbonate between the erythrocyte and the plasma. This erythrocyte, in terms of its size, is about the same diameter as the smallest capillaries. Based off of the way in which the erythrocyte is developed, it is a anucleated cell, which means that it cannot go about generating new RNA in order to repair itself if it happens to be damaged. And what's up happening is that since it's about the same size as the smallest diameter capillaries, anytime that erythrocyte comes in contact with the plasma membrane of the luminal epithelium, it's going to have small little bits of damage to the membrane. Anytime it interacts with an ROS, it's going to have small little bits of membrane damage. And as it starts to accumulate this ROS damage, and as it starts to accumulate this membrane damage, it impedes the flow through the spleen and the liver, which will lead to its recycling. Now, the erythrocyte's life cycle is approximately 120 days. So we have approximately 120 days of life from the erythrocyte. When the erythrocyte reaches the maximum amount of damage it can sustain, as it flows through the red and white pulp of the spleen, it gets trapped within the reticular regions, and macrophagic activity will start to digest it down. Same thing happens within the sinusoids of the liver. What this does is this macrophagic activity is it starts to separate the heme groups from the erythrocyte. In the heme groups, what's happening is that it will pull apart the iron from the rest of the proteins, from the rest of the hemoglobin molecule. So the globular protein molecule will get vesiculated into bilirubin to be excreted within bile within the liver, or to be transported to the kidneys to be part of ureabilirubin to be released in urine. The iron itself will be vesiculated into ferrin and then transported back to the bone marrow, particularly red bone marrow, in order to be reintroduced into the erythrocytes. This whole cycle is going to be predominantly regulated by EPO-based pathways. So what ends up happening is we start with a pluripotent stem cell. This pluripotent stem cell within the trabecular region of the epiphysium will undergo a signal for growth from EPO to form what's referred to as a myeloid stem cell, CFUE. So that's a colony-forming unit erythrocyte. So this is a cluster of stem cells which will become erythrocytes based off of secondary EPO signaling. So the CFUEs will form into pro-erythroblasts. This pro-erythroblast will undergo maturation, formally referred to as a reticulocyte. As it starts to undergo maturation as a reticulocyte, the nucleus gets ejected. This nuclear ejection is what establishes the biconcavity within the mature erythrocyte. But it also limits the ability for the erythrocyte to undergo adaptations during its own life cycle. That erythrocyte will then go into circulation for 120 days. That 120 days is why, for those of you that donate blood, you're only allowed to donate blood once every 4 months, because you need to have that time span in order to allow for enough regeneration and recycling of the erythrocytes to ensure that you haven't lost too much in terms of both erythrocytes as well as total blood volumes following the donations. So let's take a look at this cycle. And it's an EPO-regulated cycle. And so EPO is coming from the juxtarenal region of the nephron, so it's coming from just outside the glomerulus. And what it's doing is it's the only area where we have cells that are sensitive to oxygen. So regardless of how you might want to think about the responses that we see in cardiovascular and pulmonary or cardiorespiratory functions, none of it has to do with oxygen, with the exception of EPO. In these juxtaglomerular cells, they sense the level of oxygen in the renal tubules, in which if we happen to have low O2 saturation, low PO2, or what's referred to as low oxygen tension, the extraglomerular cells will go about their process of stimulating EPO release. Now, this stimulation is inhibited by high levels of lactate in circulation. If I have a high amount of anaerobic respiration going on for long periods of time, I am automatically reducing the level of oxygen saturation that takes place, which means that I don't want to go about stimulating additional erythrocytes if it happens to be an acute response due to very high exertion. However, if I happen to have moderate levels of lactate, I'll go about having this response. I go about increasing EPO production. EPO is going to head to the red bone marrow in order to stimulate the progenitor cells to undergo mitotic division and differentiation towards the CFUEs. Now, a secondary signal can come in here, and that's testosterone. So we now have part of the answer as to why males might have more erythrocytes and total blood volume than females, because males tend to have higher levels of testosterone circulation relative to females. So the combination of the EPO plus testosterone, or adrenal androgens, DHEAS, is going to lead to proliferation and differentiation of progenitor cells. This differentiation of progenitor cells now becomes metabolite dependent. It becomes metabolite dependent on two metabolites, what we refer to as the coenzymatic activity, which include iron and vitamin B, and particularly vitamin B12. And this takes these differentiating progenitor cells and allows them to start to undergo CFUE maturation, followed by reticulocyte maturation, followed by erythrocyte maturation. As we start to mature erythrocytes, release them into circulation. The release into circulation will start to increase the oxygen carrying capacity within the plasma, because we now have more hemoglobin to bind onto oxygen, which will inhibit the initial signals of low saturation and low oxygen tension being sensed by these extraglomerular cells. Now, within this cycle, we can look at two areas where endurance athletes can go about having doping issues. And we can also look at two areas where people who live in lower areas of elevation and move to higher areas of elevation will go about producing additional erythrocytes and will have larger plasma volumes as response. For the dopers, there's two areas that come into play. I can dope with EPO, thereby giving me the signal to go about doing the progenitor cell mitosis and lead to CFUE formations. Or I can dope with testosterone and get the exact same thing. Testosterone works this way because testosterone is a ubiquitous anabolic hormone. It's going to cause all tissues to grow. And if I'm causing all tissues to grow, that means I'm going to end up having more blood volume. In order to keep hematocrit stable, I need to have more erythrocytes. Now, within the dietary dependencies, we have two issues that come into play here. And the two issues that come into play deal with what I am eating within my diet, where we have to worry about distinct types of dietary restrictions causing issues that can impact anemia. Think about what type of dietary restrictions might go about limiting the ability to form the CFUEs and allowing for the reticulocyte and maturation of erythrocytes as it relates to hemoglobin formation in our codependency factors. One of the other things that we have to talk about when we talk about blood is what's referred to as the blood types. And the blood types is about the transfusibility of blood in between individuals. We utilize what's referred to as the ABO blood classifications. And the ABO blood classifications is an indication of what blood antigens happen to be there versus what blood antibodies happen to be there. And the antibodies that we're talking about are referred to as the IgM antibodies, where if we have an A-type blood, we have the A antigen but the B antibody. Where if we have the B blood type, we have the B antigen but the A antibody. If we have the AB blood type, we have the A and B antigen, but we don't have any IgMs for blood type. For the O, we have the non-A, non-B antigen, so indicated here as the none. And we have antibodies to both A and to B. Now, this blood typing registration in terms of A, B, and O classification is what's going to be utilized when we look at the compatibility of blood types as relates to transferability. That is, can I give blood and can I receive blood? And this becomes very important when we look at transfusions within hospital settings. It becomes very important when we talk about blood donations in between individuals, where we have the responses based off of immunological activation, where we have two types of blood that will not trigger and not respond to blood transfusions, where the O negative blood will not trigger an immunological response, whereas the AB positive blood will not respond to any type of blood transfusion, in which we want to make sure that we have a compatibility between blood types if we are attempting to transfuse from one individual to another individual. We'll get more into this when we look at the blood transfusion simulation when we're in lab. When we talk about blood typing, there is an indication for distinct population and subpopulation frequencies. Now, when we're talking about frequencies, what we're talking about the prominence of seeing a distinct trait within a population. It is linked to genetics within populations. However, it is not fixed within populations. So we're going to take a look here in a second at subset populations based off of blood type frequencies, where you can make a logical guess as to what type of blood the person might have based off of distinct factors for them. The most prominent blood types that we see within the United States is O positive and A positive. The most rare of the blood types are the ABs, and particularly AB negative, the B negative. Now, if you notice, the negative blood types are always less prominent than the positive blood types, and that has to deal with some of the genetics of Rh. The other thing with Rh is that Rh can be epigenetically regulated as well, and there are immunological issues that can come about for instance, an Rh mismatch in between a fetus and the mother to be during pregnancy. And so the way in which that breakdown for the whole entire population comes into play is what types of populations are the most prominent that we see within the United States. And so if you notice, the populations that are most prominent in the United States, the Caucasian, African American, Hispanic, and Asian, the most prominent of those blood types give us the indications of O and A being the most prominent blood types that are there. Now, within each of those populations, there will be subset populations where we'll have higher prominence or higher frequencies of distinct types of blood types within regions of those ethnic groups based off of origins. And we'll talk more about this when we're in lab looking at blood types.